people have connection with God, would they so?
These, in case you get a sugar need pop up. Without further ado. So, tonight, lucky you, hey, by the way, while I'm doing this introduction, run through a list of the fishes that you suspect might be in these marshes. I've got my own little list, and we'll see how they match up. So tonight, we're gonna hear from Lily Olmo, a graduate student at the university, and a recipient of a grant from FOAM, Friends of the Arcata Marsh. She's one of those brave people that puts on, it's not a waders. You can't wear a waders because I just learned they would flood if you get into the marsh. So you have to wear actual diving gear. She's been doing this for some time. She's hoping to graduate this spring, summer. And we're very much looking forward to what we can do with the work that she's done in the future. For example, we were just talking about, wouldn't it be great if there was an interpretive sign around the trails here where you could see what kinds of fishes are in the places you can't see. It's under the water that we have trouble relating to. That's where all the work happens for the wastewater treatment plant and so on. So I think it's a great idea. So I'm very much looking forward to hearing from Lily. Please welcome Lily Olmo.
Thank you, Elliot. Hi, everyone. My name is Lily, like you said, and I'm a master's student at Cal Poly Humboldt in the Department of Fisheries Biology. And these are going to be some preliminary results that I share today. I haven't finalized what's going to go in the thesis, but I'm excited to get your feedback and hear what you think about it. So before we start, I'd like to begin with a land acknowledgement to recognize that Cal Poly Humboldt and the Arcata Marsh and all of the research sites that I'm going to talk about today are located on unceded lands of the Wiyot people who have resided here from time immemorial and continue to steward these environments today. And so here is a map from Zachary Erickson. He was a previous forester with the Wiyot tribe, and he outlined the accurate oral history of Wiyot territory boundaries. And in darker yellow, inside of Humboldt Bay, you can see the current Wiyot land holdings. And what we know now as Humboldt Bay is Wiggy in the Wiyot language. So here's a short outline of what I'm going to be talking about today. First, I'll share a little bit about fish and their importance in the ecosystem. Then we'll talk about tidal wetlands and artificial wetlands, talk about the research objectives. I'll share the methods and results of the study, some takeaway points, and then I'll ask that you leave all questions till the end, and we'll have a little question and answer session. So we're going to be talking a lot about fish today, so I thought we'd begin with a fish 101. So fish are aquatic vertebrates, which means that they do have a backbone, and they use gills to breathe, and instead of limbs, they have fins. So this is our favorite salmon, and you can see some of the features on it, particularly the gill plate or operculum. I forgot I have a pointer. Right here, which is where the fish takes in water and extracts oxygen. And then there's a variety of fins also, where the fish is going to use those to steer and swim through the water. And I also wanted to point out the lateral line that most fish have, which is a set of specialized cells that allows them to sense vibration so they can move or avoid predators. But fish have evolved over 530 million years, with the first fish in existence looking something like this. So a lot more basic than some of the fish that we might think about today. And so now we have over 36,400 species of fish in the world and counting as we do more research. So let's bring it into Humboldt Bay where we sit right now. These are the 6 unique watersheds colored that enter Humboldt Bay that bring some fresh water. But the majority of water in Humboldt Bay is pretty marine, and that's coming from this area between the north and the south spit where ocean water is entering. So that creates our estuary. We have brackish water here, which is that mix of fresh and salt water. And this is a great refuge and nursery for many different types of fish species. In Humboldt Bay, we have over 120 species that live here at some part of the year, and 50 of them live here year-round. So some species are just migrating between the ocean and the bay, or the rivers and the bay, which is why they're kind of partial residents. So why is it important for us to study and understand the fish that live here? I'm going to give you a variety of reasons here. Also, here are some species that you might be familiar with. So on the top, we have the northern anchovy and a surf perch, all the way up to California halibut and even leopard sharks, which are considered fish. So why study fish? The primary one that this study was motivated by is ecological value. Fish can be great indicators of overall ecosystem health. So what I mean by that is, let's say we have this healthy wetland and we assume there's lots of good food for fish and we have enough oxygen and the water quality is good. So we might expect to see fish here, large fish of all sizes, and in abundance. But if we think about this dramatic example of a polluted wetland ecosystem, we might not see as many fish, maybe smaller fish that are in poor health, and they might even be dead from the different fissions. So by studying fish in all different types of ecosystems, we can start to get a gauge or a pulse for the health of the ecosystem, which is why they can be called indicators. They're also an important component of complex food webs. So here we have a salt marsh diagram, and this is very simplified, but we have our primary producers on the bottom level, like grasses and plankton, and then that comes up to our primary producers and then secondary and tertiary consumers. And what you can see is that fish are involved in multiple parts of this web. So here they're primary consumers as larvae and juveniles, but they're also secondary consumers and tertiary consumers, sometimes even the top level predator. So they're this critical link in between predators and prey. Next, we have economic value. Fish are an important industry, especially here on the North Coast, for commercial fishing and recreational fishing. And fish are also contributing to a vibrant tourism industry, as well as the value that they serve as food and protein for a big majority of the human population. And this is a graphic from the National Oceanic and Atmospheric Administration, and this data was pulled from 2020. And it includes data from both commercial and recreational fishing, showing that it provides 1.7 million jobs, 253 billion in sales, and 117 billion in other services. And this doesn't even count the value that fish have that we can't directly calculate. So this is a fun trivia question because I did not know this statistic and I thought it was pretty cool. What percent of the entire state's entire seafood harvest do you think comes from the North Coast specifically? And this is like mainly commercially. Any guesses?
Yeah, we have an upwelling zone, so we have a lot of fish, right?
Yeah, well,
20%.
it's 26%, which is still like higher than I thought that it would be, just because we're like a smaller region up here than some like SoCal. And then for our North Coast region, how much do you think that these harvests are worth annually? Any guesses? About 40 million. So it's a pretty significant economic driver in this region. And the fish in our bay are directly contributing to this productivity. I also wanted to mention the cultural value of fish, particularly in this region. It's important to the recreation and leisure of a lot of people, but of particular importance to our tribal nations, both here on the North Coast and across the country, who rely on these foods not only as subsistence food resources, but these fish are also an integral part of culture as well. Another reason we might want to study fish is because we live in a changing world and our ecosystems are changing. So let's take Humboldt Bay. It's experiencing the highest rate of sea level rise in all of California. So this is a map of Humboldt Bay, and the blue areas show the current sea level, and the dark blue areas show what the sea level will be with 3 feet of sea level rise. So you can see that the area that we're in right now of the marsh is part of that dark blue zone. and this rise could happen around 2060. So this threatens our human communities, but also could influence what fish are living in which areas of the bay as well. Another change we're experiencing globally is climate change, which definitely impacts the health of our oceans. And this graph or this figure shows the annual sea surface temperature anomaly. So an anomaly is how different the temperature is right now compared to past conditions. So this is showing the data from 2023 compared to the 30 years prior to that. So any red areas mean that the temperature overall has increased, and then the blue are areas where temperatures have decreased. So in these dark red areas, like in the Pacific Ocean, you can see up to 2 degrees Celsius of increase in temperatures. So fish have optimal temperatures that they can live in or even thrive in. So it's important for us to know how these changes are going to impact our bay system as well. Another change occurring here locally is the development of the bay for aquaculture and offshore wind. And I think there are a lot of conversations going on about how we can do this sustainably and minimize our impact to aquatic organisms. But we also just don't have a lot of data about what's under the water. And so there may be some unanticipated changes. And how can we know if we don't have data before? One last reason here for changing ecosystems are invasive species. These are species that are not native to our watersheds that were brought in. So this example of the Sacramento pike minnow in the Eel River is a good one. They came in the 80s to the Eel River, and they compete with native salmonids, and they can even eat the eggs or the juveniles here. So there's been a lot of work in trying to rebalance these ecosystems, and the introduction of new invasive species is still a concern for Humboldt Bay. And lastly, I think it's important to study fish because we don't know a lot about them and they remain a mystery underwater. There's much left to be explored about their biology and their ecology, especially in this changing world. And that's what I get excited about. And that's why I am here today. So I grew up in Ohio, not thinking much about fish, but when I went to school in North Carolina at Chapel Hill, I got involved in a salt marsh research project looking at fish communities. I even worked on sea turtles in San Diego Bay, and then I had the opportunity to work at different federal agencies and a tribal nonprofit looking at sturgeon and salmon and halibut populations. So I really gained this interest for human communities and fish communities as well. And I found that I really enjoyed studying estuaries because they're this dynamic spot in between fresh and saltwater, and we have such a direct impact on them. So I want to introduce tidal wetlands or tidal marshes, which are the habitats that we're going to be talking about today. So on the top, we have a aerial photo of Humboldt Bay. And on the bottom, this is what these fringing habitats around the bay look like. So they're exposed to tidal influence. So water's coming in and out with the tides. Some services to us, which are any environment. They're really good at nutrient cycling. They store water, and so they can help us with erosion and flooding protection. They sequester carbon, so there's a lot of work going on to see how we can suck out carbon out of the atmosphere and get them into the sediments of the marsh, because that's a natural function that they do anyways. And they can be great habitat for fish and wildlife, and particularly for fish that we're going to be talking about today. These areas serve as foraging grounds for feeding, spawning, nursing, and even spots to migrate through. So we have a lot of reasons to protect these habitats. But this is the global status of wetlands. This is wetland loss from 1700 to 2020. And the areas that are blue have the least percent of wetlands lost. The areas that are red have over 75% of wetlands lost. It's a little hard to see, but here on California, we are in that 75+% of our wetlands that have been lost. And in California, that statistic is 90%. And so in Humboldt Bay, we track the statewide trends. We have less than 10% of our wetlands remaining. So you can see some of these pictures of the development of the railroad and agriculture, the marinas, and even modern day development coupled with erosion and sea level rise. All of these threats make tidal marshes vulnerable because they can't sustain their function if they're underwater. So there's been an uptick in interest in restoring these habitats and even creating new artificial or human-made wetlands to take their place. So artificial wetlands are defined a little bit differently depending on what you read, but for my purposes, I'm defining them as human-made wetland systems. So there's a variety of reasons that you might create a wetland. One example here in Arcata is the wastewater treatment plant. We might also create wetlands for habitat restoration. They're an important part of aquaculture and agriculture industries. We can use them as water storage, and we can also use them as water filtration. So maybe none of these were created for the purpose of fish, but they end up creating fish habitat regardless. So that's where the Arcata Marsh comes in. So as many of you know, this system was created as an environmentally efficient wastewater treatment system. And there are two ponds that I'm going to talk about today that are tidally influenced, which are Clop Lake and Brackish Pond. And if you came to Andrew Wolf's presentation last month, he had a list of people's most reported sightings at the marsh, and otters and birds were obviously super high up. And then like halfway down the list were fish. So hopefully you'll learn something here today and keep an eye out next time you're walking around or thinking about the marsh. So if you walk the trails here, here's just to orient you. We've got Brackish Pond up here with the yellow arrow, and then Clop Lake down here. This is an aerial satellite image of the marsh area and then downtown Arcata in 1854. And this is what that looks like today. And you can see that we now have these wetland ponds in areas where wetlands used to exist, but also in areas where wetlands didn't used to exist, like on land. So these are very different than natural wetlands because they're full of water 24/7. All the natural wetlands around it are draining with the low tide and filling with the high tide. So this creates a really interesting habitat for us to study. I'm also going to be talking about one site that I studied at the Humboldt Bay National Wildlife Refuge, and this is south of Eureka in Lolita. So this is Long Pond. If you've ever walked the Shorebird Loop Trail, you're familiar with this pond. And it was created in the 40s over old agricultural land, and it's connected to South Humboldt Bay. So it provides an interesting comparison for us to compare to the Arcata Marsh. So in all, we've got our two sites in the North Bay, Brackish Pond and Clop Lake, and one site in the South Bay, Long Pond. Obviously, there are more of these ponds, but these are just the three I chose to focus in on. So what exactly am I trying to do here? I am interested in looking at the entire fish community and how that changes over the seasons. So I had a 10-month field season that went from February to November of 2024. And my specific questions were what environmental conditions are present in these ponds, which fish live in the ponds and how many, which fish are moving in and out of these ponds with the tides, and lastly, which life stages of fish live in these ponds. So by life stage, I mean larvae or juvenile or adult. So the first thing I knew about this ambitious project was that I was going to need a lot of help to do it with all the different field methods. So I was able to hire 3 undergraduate technicians who helped me with this project, and the Friends of the Arcata Marsh grant really helped with this. We were able to have gear for all of the technicians that they're able to use for our long field days, and waders, wetsuits, boots, all of that, and they're able to use this gear for future job opportunities that might require gear, as that can be like a pretty big barrier to getting a job in this field. So if nothing else, the project was already a major success in this regard. So coming to our first question, yeah, that's me in Long Pond. What environmental conditions are present? So we're going to collect some environmental data. I was able to read through some old reports, look at satellite imagery to get the dimensions of these ponds. We took field observations every time we went out to sample fish. And primarily we're looking at water quality and measuring the environmental factors within the water. So salinity, temperature, dissolved oxygen, turbidity, and water height. So I'm going to walk you through each pond and what we found. So this is Brackish Pond. It was created as part of the Arcata Marsh system. and it is the farthest away from the mouth of Humboldt Bay. It's characterized mainly by mud with some grass and rocks when you look under the surface at the sediment. And we regularly saw pelicans and wading birds and otters here. So Brackish Pond is connected to Humboldt Bay through McDaniel Slough with this large metal pipe that you see here on the right. And that's where the tidal water can come in and out. And then on the opposite corner with the new wastewater treatment upgrade, wastewater effluent that's all treated started going into the opposite corner of Brackish Pond, and that's what that looks like coming out. And this happened in November 2023, so before any of my sampling began. Next, we have Clop Lake. This is the largest pond or the medium-sized pond in our study, but the most visited spot in the marsh. And the bottom here was also mud and rock, but we also had a lot of eelgrass patches and different shells like mussels and clams. We also saw otters here the most, marine birds and wading birds. And this is where Clop Lake is connected to Butcher Slough. And so you can see us at the structure. That's where the water is coming in and out of the bay. And what we realized pretty early on is that this pipe is pretty collapsed. And so we weren't sure if fish were going to be moving in and out of here, but we did know that some water was at least making it in and out. Last, we have Long Pond, which is in the Salmon Creek unit of the refuge, and this is by far the largest pond in the study, bit of a different shape as well, and it's closest to the mouth of Humboldt Bay. And the bottom is mainly mud here, as you saw in that other picture. And we also saw otters and birds here. So in Long Pond, instead of a pipe, we have this large tidal levee. This thing is over 20 feet tall, so much larger than any of the structures at the marsh. I just don't have a great photo of it. But there are a couple areas where fresh water also comes into this pond through Salmon Creek. The other thing is that this site ended up closing down halfway through our season because they had to repair the tidal gate. So past June or July, when I show my data, I won't have anything for Long Pond past June or July. So here is one of my technicians taking water quality samples with a probe. And we did this every time that we sampled fish with any method for the whole season. And then we did so in each of the corners of the pond, so we could capture some of the variability that might exist between these corners. So I'm going to show you what that data looks like over time. So for example, we have salinity here. Salinity is a measure of how salty the water is, how much salt there is per 1,000 particles of water. So you see here salinity going up here, and then we have our time on the bottom. Each pond is a different color and shape for the next few graphs. And then this red dashed line is the salinity of the ocean. So what I noticed here first is that Clop Lake is our saltiest pond, and it even exceeds the salinity of the ocean at times, which at first I was confused, but it makes sense if this salt water is coming in, water's evaporating, it's leaving behind concentrated salt, and it's just not super well mixed. And then we have these other ponds which can have fresh water all the way up to saltwater measurements. So it's important for us to know these things because fish might have different tolerances to salt and have preferences. We also have temperature here. So temperature ranging on the y-axis, and then our red line is the average temperature of the ocean. So our ponds are going to be warmer than the ocean because they're not getting as much mixing. And in general, we see what we would expect with temperatures increasing from February to July and August, and then September to December, we see this decrease. And the one thing that stood out to me here was that Brackish Pond in green, in February, March, April, the colder months, it is the coldest pond, but then in July, August, September, it's the hottest pond. And so it's experiencing the most variability. It is the most shallow, so it's going to pick up heat quicker and release heat quicker as well. And again, fish might have temperature preferences, so this information is good to know before we start looking at the fish. This is dissolved oxygen, so the level of water that is in the water, and fish are able to extract that oxygen, so we don't want to see any concerningly low values of DO, which we didn't. We see pretty variable and actually pretty high DO. So over 100% means that it's super saturated with oxygen, and it's pretty variable. There's a lot of reasons for this. Early in the morning, DO is a little bit lower, but as plants photosynthesize in the day, they're increasing the DO. And so because we took measurements at different times of day, we weren't necessarily standardizing for that. And mixing within the pond and how much the water is getting kicked up can also influence DO. And then the last environmental graph is turbidity. This is a measure of clarity of the water. So on the y-axis, this would be really clear water all the way up to really muddy water. And generally, these wetlands are going to have pretty high turbidity because the mud is getting kicked up into the water. This can affect fish because they need to be able to see in the water for feeding and navigating, and different fish have different tolerances for that. So we see a lot of variability here, and in general, all of these environmental trends are just helping us to understand what fish might be able to live here. Which kind of goes into our second question, is which fish live here and which ponds do they choose to live in and how many of them? So for this method, we're using seine nets. This is this large nylon net. This one's 100 feet long. And so you can see one person is on the shore and then one person's dragging the net out, and we just do it that way into a large circle. And then at the end, we circle it up and close it. There's a lead line at the bottom, which weighs the net down so that any fish can't escape from underneath it. And then we pull it up onto shore. So we'll close the net and then we'll bring all the fish into the bucket, and we'll use different identification guides to figure out what fish we're looking at. We can look at the position of the mouth or the position of the fins or the size to try to understand and figure out which species it is. And then we'll measure the fish as well, and that will help us get an idea of its age as well as what it could weigh. So we'll release all the fish back into the pond, and then we'll go to a different corner and do it again, and we'll have 4 every month for each pond. So one day, one of my technicians and I were thinking, why don't we just do like a census? It would be a lot easier. We could just like snorkel around and ask them, who are you and where do you live? That would be a lot easier, right? But that's essentially what we're doing with all these different methods, is trying to get a census of the fish community here. So in total, over our season, we had 10 months for 100 samples, and we captured over 6,000 fish. Each pond here is a different color next to each other, and then we have the total catch, or the total count of fish that we got that day. So what we can see here, start to see here, are some seasonal trends. So Long Pond has a high abundance in May and June. We don't have data past that, but it could have been higher. We also see peaks in Brackish Pond in June, July, and August, whereas our peak for Clop Lake is in May and November. But looking more specifically, we can break this down by pond, and instead of saying total count, we can look at it by species and say how much of this sampling, or how much of these species are making up these samples. So you can see that now we see the same peaks in June and July, but now we can see which species are contributing to that. So in June, this is Brackish Pond, we have tidewater goby is the majority, followed by some 3-spine sticklebacks. And then we have 3-spine sticklebacks and top-smelt silversides, which contributed to those peaks. And in total, we captured 9 unique species. The color represented on this graph is kind of an indicator that it's a pretty diverse system. And this is many more species than we thought that we might get in a human-made pond. So in total for Brackish Pond, we captured over 1,300 individuals with our 40 samples. So I'm going to bring out our podium and talk about some of those species that were the most common. So in third place, we have the top-smelt silverside. This fish was 20% of our sampling in Brackish Pond. And these fish are not true smelt, even though it has smelt in the name, so that's very confusing. So they're not in the night smelt and surf smelt family. They're a small schooling fish, so they'll swim in groups together. And in Brackish Pond, when we were getting top smelts, it was primarily juveniles. So juveniles move from freshwater to saltier water as they age, and Brackish Pond kind of has both conditions, with freshwater on one corner with the effluent, and then we have salty water on the other corner coming in from the bay. In second place, we have the tidewater goby. This was 22% of our samples, and these fish are fairly small as well. They have a sucking disc on the underside of their body, which helps them to prop up on the bottom of these ponds, and they're a bit more sedentary than something like the top smelt. And these tidewater gobies are a federally endangered species. So in the 90s, the US Fish and Wildlife Service made it a priority to designate some critical habitats for these guys and also try to rebuild their populations. So that work has been going on in Humboldt Bay, and there had been some records of tidewater goby in Brackish Pond, but no consistent sampling. And so we showed that they were there all 10 months, even after the implementation of this effluent as well. And then in first place, we have the three-spined stickleback. This accounted for half of our entire catch in the pond. And they're also no more than a couple inches, and they're named because they have 3 spines on their back to prevent predators from eating them. They can tolerate a wide variety of conditions. So these are one of the most common fish that we get in any estuary sampling because they're so tolerant of the variations that come with being in a tidal system. We always joke in our department that we hate these fish because imagine getting a bucket of like 4,000 of these that you have to count, and you might be a little upset too. But I do have a special place in my heart for these guys. And then our honorable mention for Brackish Pond is the Pacific staghorn sculpin. It also has 2 little antlers that deter predation, and it made up about 7% of our samples. They'll hang out on the muddy bottoms of the pond, and they can actually breathe when they're out of water, which other fish cannot do. This is Clop Lake, so you'll notice first the colors are pretty much all different. So new species that we're looking at here. We had 7 unique species in total, and we can see that the peaks in May were dominated by yellowfin goby and 3-spine stickleback, and then June, also yellowfin goby. We had a movement of top smelts in November as well. So a bit less abundance overall, a bit less diversity overall compared to Brackish Pond. And we had 794 individuals. We had 10% sticklebacks, 22% top smelts, and then 44% yellowfin gobies. So yellowfin gobies are an invasive species to our area. They were originally in Eastern Asia and ended up in the San Francisco Bay in the 70s. And since then, in the last several years, really, they've made their way up to Humboldt Bay. There have been a couple, sporadic sightings of them, but we've really found a hotspot here with Clop Lake. And these gobies are much larger than any of our native gobies. They can grow up to about 1 foot long and live for 3 years. The benefit is that the otters seem to love these gobies. So every time that we were out in the field and we saw otters feeding, it was almost always yellowfin goby. And our honorable mention for Clop Lake is the bay pipefish. This made up 8% of our catch. These fish live in eelgrass habitats, and Clop Lake was our only pond that was salty enough to have eelgrass. So it was really cool to see those guys in there. Then we have Long Pond. Overall, we had 4,000 fish. It was so many more fish than any of our other sites, but we didn't even have a 3rd or 2nd place. We just have a 1st place because it was 99% sticklebacks. So they really dominated this pond. Not a lot of diversity here. We did catch 5 species in total, and you just really can't see the impact of those because they're so tiny. And who knows what we would have got if we were able to continue sampling the entire 10 months. Long Pond is interesting because of that tide gate. They manage it depending on conditions. So sometimes that gate might be closed and sometimes it's open. And maybe these fish just aren't able to deal with that variability. So when I asked the question, how many fish live in these ponds, we've been talking about counts and the actual number of fish, but we can also think about it in terms of biomass or weight. So in the field, we're measuring our fish. We measure to the nearest millimeter, and then we can use equations that are already published online that relate the length of a fish to its weight. So if I know the length, I can then calculate a weight for every fish in my sample based on what species it is, and that way I have a full data set of weights as well. So one of the motivations for understanding weights or biomass is because of this fish's role in the greater ecosystem. And for example, we have a sculpin. it's eating and accumulating biomass with fish eggs and invertebrates, but then it's also important biomass for river otters and other fish and marine birds like cormorants. So by calculating biomass, we're also getting a relative idea of fish as food and energy. And here are a few local highlights from some local photographers. Some of you are in the room, and you can see maybe that these are all yellowfin gobies that they're eating. And this is a picture that I took on the right. This is where Brackish Pond enters the slough, and it's hard to see, but you can see heron on the left, heron on the right, and they're just waiting for that rush of fish to come out. It reminds me when you're at the sushi restaurant and the little train is coming by and you're just like grabbing as you go. They're literally just waiting for these fish to flow by. So every time we went out, there would be some fish posted up there. Another reason we might study biomass instead of counts is because it tells us a bit of a different story. So here we have 1,000 stickleback, which are generally small fish, that maybe would total to 1,000 grams. But we could have 200 gobies that bring us that same amount of biomass. So again, it's telling us a little bit of a different story in terms of the food web. So this is the same graph that I showed you earlier of just counts for each pond. So let's zoom into like July and August, and you'll see that, again, Brackish Pond had the higher abundance compared to Clop Lake. So if we focus on that area of the graph, I'm going to transform these counts into biomass. And so what you'll see is that the trend is a bit reversed, where we have more biomass in Clop Lake. So over the sampling season, when I totaled it up just for fun, we sampled 1.2 tons of little fish. Then I wanted to know, okay, so we see these differences in biomass, which species are contributing the most to that biomass? So this is a percent composition graph. So on the Y, you have the percentage of the total biomass that was attributed to that species. So for Brackish Pond, we see a couple of main species that are contributing to the overall biomass in the whole year. So we have top smelts, we have sticklebacks, tidewater gobies, and sculpin. Then you look at Clop Lake, it's more dominated by the biomass of yellowfin goby and top smelts. And then in Long Pond, again, we do see mainly sticklebacks, but you see some of those other species showing up a little bit more. So we can start to see what these otters or birds might be eating by looking at this, and still more work to be done here. So our third question, which fish are moving in and out of these ponds? We use fyke nets. These are a stationary net. This is a diagram of one. So you have the main frame, and then you have some wings that go out to the side. Basically, all the fish are getting funneled back this way into these rings. So for example, we have this net here. If the water's flowing out of the pond, all the fish are going to get trapped in this back section and we can sample them. So we would do this on an outgoing tide. We would leave it for an hour, see what fish we got, and then when the tide flipped, we would just rotate the net 180 degrees and then do an incoming tide as well. So this is kind of what the setup looked like in each of these ponds. This is Brackish Pond, right outside the pond. We couldn't get it right up in the pipe, but you can see that all the water flow and the fish that would be moving are constrained by these wings and getting caught in here. This is Clop Lake. We can put it kind of right in this pipe wall. And then Long Pond, I don't have a great picture of the setup that we did here, but it's really deep right here, so we did have to set it like farther back to be able to catch anything. So this is counts, and on the top we have the species that we captured on any incoming samples and the species on average that we captured on an outgoing flow. So for Brackish Pond, we see a few more fish coming in than out. In Clop Lake, we really don't see many fish moving. Again, this is the one where the pipe is collapsed, so I really don't think that fish are moving through here. And instead that this little yellow green bar that we do see is actually just fish that are in the area swimming around and end up in the net. But if they were actually trying to move through, they wouldn't have been able to. And then we have Long Pond, which had a lot of sticklebacks coming in, maybe less going out. So also still more work to be done here to split this up and look at it in a bit more depth, but we can start looking at fish movement as well. Another benefit of the fyke net method is that we caught all of these species that we didn't detect with our other methods. So When we're seining, fish are, especially fast swimming fish, are able to avoid our net. But with pike nets, these fish are funneled in with the pipes and they get stuck in the pike nets. So that's probably why we caught some of these fish here. So we have a surf perch. We had a large movement of adult top smelt that we didn't get in our scenes. We got marine species like Pacific herring and northern anchovies, mainly in their larval or juvenile phases, and then longfin smelt, which is the first ever recording of a longfin smelt in the Arcata Marsh. This is a threatened species, so that was pretty exciting to see. Lastly, we have which life stages of fish live in these ponds. So with our other methods, seine and pike nets, we got a good idea about juveniles and we got a good idea about adults. But what we were missing were larvae, or the earliest life stages of fish. These are really tiny and you can't sample them with the other methods. So we have to construct these handmade traps that my lab helped me construct. And they have an LED light strip in the middle, and this attracts larvae that are attracted to light. They come in through the funnels. We set it in the evening, and then when we come back in the morning, the fish will have funneled in there and we can sample what we've got. So here on the left is what one sample from one trap looks like. So there's hundreds of fish in there and they're really hard to identify. So we use a dissecting scope to be able to see the features. But even then, fish are changing their features as they grow from larvae to juvenile to adult. So it can be really difficult and timely to identify these fish. So I don't have all of the larval samples processed yet, but I'll share what I have preliminarily. So for Brackish Pond, this had the most larvae. It was primarily anchovy and tidewater goby. And this is an anchovy, just that part of its body is 10 millimeters long. This is Clop Lake, which primarily had yellowfin goby, pipefish larvae, and top smelts. And these are 14 and 15 millimeter pipefish. Adorable. And then we have Long Pond. We only got a couple dozen larvae here as opposed to thousands of larvae in our other ponds. And so likely this tidal gate might restrict the movement of new fish into the pond. And this is a staghorn sculp, and you can even see its spines here at 28 millimeters. There are certain fish which are incredibly hard to identify, like this one, or they might be damaged. So this fish is only 3 millimeters. It still has its yolk sac, which it's feeding off of. And I can't find any characterizing features that would say this is definitely this fish. So one thing that we're able to do is take eye tissue or fin tissue and extract the DNA. And the DNA can be compared to a reference database that has all species in it. And we can figure out that this 3 millimeter dot is a yellowfin goby that was probably just born in Clop Lake. So this is just a really cool addition to what we are able to do to improve the accuracy of our identifications. And I want to thank Michelle Scheidemann and Andrew Kinzinger for helping me with this part of the project and helping to run these samples. We also got species in our light trap that we didn't get in any other methods. So this is a plainfin midshipman, and this is a marine fish that has small pores that you can see in that bottom photo that emit light, and they can also sing. So they bury themselves in the dark, and they're not, or in the mud, and they're nocturnal. So that's maybe why we didn't see them during the day. And then we also had one observation of a saddleback gunnel. This is what it looks like as an adult. This is the larvae version in Long Pond. So we definitely didn't expect to see these guys, and that was pretty exciting, adding more to our understanding of which fish are even here in the first place. I also wanted to highlight a couple instances of spawning and reproduction that we also saw. So this is a top smelt silverside. It's fairly small, but there are some small yellow specks on the scales, which indicates that this fish is breeding. This is also a stickleback, and if you know what it's supposed to look like, this is really large, so pregnant female, and this species is cool. The males nest, they guard the nests, and so they'll build a nest and they'll guard the eggs and the young. And then we have the pipefish. This enlarged section up here is actually a sac full of larvae. They give live birth, and so just like seahorses, the males are the ones to carry the babies. And we observed births happening in Clop Lake a couple of times. So some take-home points here. Artificial wetlands are providing habitat for diverse fish communities in Humboldt Bay. Our counts in biomass are driven by particular species, and these species differences are likely due to the differences in the ponds, their construction, and their environmental conditions, as well as the design of those gates and how much water they're allowing to enter. So what does this mean for our area? So now we have an increased set of data on the who, when, and why of fish in these artificial wetlands. Like I talked about some of the values of fish in the beginning, we need to be able to understand them to sustain these services, to conserve these fish. And it's particularly important in the face of some of these changes where we don't know what's going to happen. We're creating baseline data before these ecosystems might change some more. I also think we highlighted the importance of a multi-method survey approach. So if we had used just one of these methods, we wouldn't have gotten the full story. So by adding these methods together, we got a much more complete picture of the species that were here. And we recorded 13 species in ponds that they were not previously recorded. So 6 species that we didn't know were in Clop Lake, 6 in Brackish Pond, and 4 in Long Pond. So this will be really important information for whoever continues to monitor these ponds in the future. And I hope that people can expand this sampling to more sites because there are many other tidal wetland ponds that have not had any fish sampling, so it'd be really interesting to compare those as well. And ultimately, the dream would be to inform the design of future wetlands for fish. So is there a way that we can design these wetlands with fish in mind? Because that's usually not the primary objective when these ponds are created. So my next steps for this project, I'll be finishing analyzing all the data. That'll include statistically linking the fish data to environmental data in a more quantitative way. And then I'm trying to get hold of some old sampling records to be able to compare to any past sampling that has been done in these ponds. I'm hoping to give a walk at the marsh this spring as part of the foam grant. And I was talking with Elliot about a panel for fish because people would always ask us, there's fish in this pond? Or like, what fish are here? People were generally really surprised. So I think that's a cool opportunity to educate people now that we know what's in there. And then I'll be defending my thesis this summer, hoping to publish this research. And I'm very excited because I'll be continuing this type of research with a PhD at Oregon State University. And there I'll be studying tidal wetland ponds, but in Hawaii, which is really exciting because this type of work can transfer to those same systems. Here's some of my favorite pictures of fish that we got. Again, lots of small fish, but we see such a wide diversity of fish, and many people at first sight might not think of the importance of these fish, but we think about their greater role in the ecosystem and how they contribute to the food web and the marsh that we love so much. And these are some other exciting critters that we found in the water. We even got a jellyfish in our nets once. This is a sea squirt. We got frogs, mussels, clams, shrimp, birds, and of course the otters. So we were always finding something new, and it was fun to track like the seasons that we were going out in and seeing the changes of the marsh every month. So first, I want to acknowledge the field technicians and volunteers that made this project possible. We had super long field days. We were in the mud, we were in all weather, we got hailed on. So it was really exciting to see their excitement for learning new things and contributing to this research. And we also held a seining workshop for the wildlife club on campus as well to teach them how to sample fish. And I have so many other people to thank that have helped with this process from the very beginning and have provided their time in the field or their advice to this project, as well as all the people who showed interest in funding this work. It was really exciting to see how many people are excited with this type of work and willing to support it as well. So with that, I would like to open up for some questions. And I also have my email up here in case you have to leave. I have business cards as well if you want my email. I love to chat about this stuff, so please feel free to reach out.
University has a committee that you have to go through, but what about coastal extension?
Yeah, so the approval process, I spent my whole first semester on the approval process. The first permit was the California Department of Fish and Wildlife permit. And then we also have an ethical use permit that we need to get to say that we're going to do this work ethically and not harm fish. And then I also had to get a permit from the city to be able to access their sites. And then the refuge is also a different permit system that allows us to access the refuge.
Wow.
So yeah, it is a lot of work writing the permits and getting that approval. It can take like 3 months. So yeah.
Congratulations.
Yes.
Survived the paperwork.
Yes.
Well, what's your favorite and least favorite part of the
Ooh. I think my favorite part was probably just that every day was so different and we were always finding new things that we didn't see before. And that was really exciting to kind of feel like a detective in that way. Least favorite part, I think, it's challenging to work in the fields in all types of conditions and like keep morale and be excited about the work regardless. So yeah, I mean, there are days when like you're sore and you don't want to be out there, or it's hailing and we were out there and we were already starting our sampling, so we just had to finish. So yeah. Yes.
Why was there more fish in the summertime than in
Yeah, so in general, fish are in higher abundance in the summer because we have more productivity, more sunlight, more nutrients in the system. That's also when these fish are spawning as well. And so the populations are blooming at that time of the year because they have more food resources and the weather's better. Yes.
Do you know anything about someone studying whether the rerouting of the wastewater treatment into Brackish Pond was going to produce any changes in the fish populations?
Unfortunately, we don't have any much data before this pipe went in. And so all of my data is after that point. It doesn't seem to be, I mean, that is our most diverse pond, at least of the ones that we did study. What's happening is that on one end, on the bay side, you have a really salty corner, and with the effluent, it's more freshwater. So the types of species you see on one corner are very different than the other corner. And maybe that's what makes it more diverse. So we might see freshwater species here, marine species here. Yes.
What was your most exciting discovery of the plastics? Well, like the fish species you found and all the unexpected places you found. What was, kind of your head?
I think we were out at Brackish Pond once. I showed the picture of the large adult top smelt. We had mainly been getting juvenile top smelts for the entire project, but there was this one day we had to make up a sample. I wasn't really feeling it, and we put the pike net into the water, and we came back an hour later after our sample, and one of the technicians was like, "Oh my gosh, this is so heavy. What is this?" And I was like, "Oh my God, there's a really big fish," but it was just a bunch of like big top smelts. So it was exciting to see that because obviously the top smelts are having to get in there somewhere. So these adults were probably coming into the pond to breed.
I'm amazed at how complex what's going on, like just in the two marsh ponds. I mean, the diversity of species and everything about it, and it warrants more than one interpretive sign. That's what
Yeah.
there's a lot going on. I
Yeah,
appreciate all your work too.
thank you. Yeah, I really would like to do the interpretive panel. Apparently there's some city approvals involved in that process, but I think that would be really cool. So if anyone knows how to use like the software that you use to produce these beautiful illustrations, talk to me. Because I haven't done something like that before.
In person that we work with. Yeah.
Improve fish passage into Clock Lake.
So
It was collapsed. Right, the tunnel.
yeah, in Clop Lake, that collapsed pipe. I think that collapsed a little bit back during an earthquake, and they're still in conversation about what to do with that pond. It's kind of interesting because there are a lot of invasive species in there. Like, do we let them out? Do we want to bring back in the flow of the salt water to increase the mixing in the pond? So, the city will be deciding like what their objectives and what their priorities are, what to do with that pond.
It's just called over your fever.
Yeah.
Does the city have a method or a program for opening the pipes to each of these lakes?
It seemed, so like at the refuge, it was very sporadic. It was based on rainfall levels. So if the water level was really high and the pond was already really full, they don't want any more salt water coming in. So they're going to close off the gate. So that was the main motivation there. Clop Lake is usually, I think, open. It's just that it collapsed. And then Brackish Pond, I think also during the higher flows of the year, they have the flap to that pipe partially closed. And then in the summer, when it's drier, they open that up. So I think a lot of it comes down to like how much water these ponds can hold.
So it's more of a seasonal operation than daily.
Yeah, yeah.
Can you tell us about that stickleback in the south part of the bay and why they're so predominant? Was it 99% of your sample?
Yeah.
What about their life cycle or style accounts for that?
Like I said, they're very tolerant to different ranges in salinity, like a much wider range of salinity and temperatures than some of these other species. So they're like more hardy, a little bit less sensitive. With Long Pond, that pond would completely drain at certain times. So there was very little water in the pond at all. So maybe not as continuous fish habitat as we thought. So I think part of that in and out movement being restricted probably limits which species can actually live there. And so the most tolerant ones are probably the ones that are going to be able to survive that.
Okay. Were you surprised that there were no salmonids in your sample, or are they not really commonly humble?
I was curious about that. They're definitely moving through the bay. They're moving even through the sloughs, but they're not necessarily going out of their way to come into the ponds. We didn't capture any. I thought maybe we would get one or two because Salmon Creek does drain into Long Pond. But yeah, they're probably just like straight tracking it for the freshwater or going out to the ocean, whatever they're up to.
So are there any potential next year's grad students that are gonna pick up the baton and because it would be really sad to just have this stop.
Yeah, no, I would love to see this work continue. I don't know if there's any current plans for that. I think the biggest hurdle always for these kind of projects is funding, and writing grants to get funding to do the field work and to hire technicians. And yeah, I think, being able to pass this on to somebody else and like continue this would be awesome.
Well, after you get your PhD, you can come here and teach and supervise.
Yes.
It's a super cool project and congrats on the PhD. I was a little surprised that there weren't any flatfish in your species richness charts. And I just wanted to ask if you had any ideas on the synergy of flatfish.
Yeah, I'm wondering partially if that's because the pipes are raised up from the bottom of the outside of the pond, so they're maybe not, swimming up to get into that. But we did get a picture from Mark Larson, who's right here, and he showed a starry flounder, an otter eating a starry flounder in Brackish Pond. So we didn't observe any flatfish, but yeah, it's very possible that there are even more flatfish in there. They might also be in the deeper parts of the pond that we couldn't access because it was too deep. So yeah.
Any more questions? All right, well, thank you very much.
Thank you, everybody. I'll turn this off here.
Excellent, excellent job. Thank you.
Yeah, what intrigued me there was that